This is a lab demonstration for the enzyme lactase laboratory summative assessment. In this laboratory investigation, you will be manipulating a variable and measuring the rate of the lactase enzyme activity. This is a video to show you an experimental setup that you can change to influence how productive your enzyme is. Here's the experimental setup. I start with 2 test tubes. Each test tube is filled with 10 milliliters of milk. Milk contains the sugar lactose, a disaccharide, 2 monosaccharides bonded together. Those 2 monosaccharides are glucose and fructose. To one of my tubes, I add something else. This is a pill called Lactase. People take this pill who have difficulty digesting dairy. Dairy contains the sugar lactose, just like this. I crushed up that pill in a mortar and pestle, and I dissolved it in water, forming a solution. I took 2 milliliters of this solution and I dropped it in one of my tubes. That's why you can see one has a little more than the other. Then I set a timer for 3 minutes. These lactate pills contain an enzyme called lactase. Lactase breaks down lactose, the substrate, into 2 products, glucose and fructose. There is a picture of lactose, glucose, and fructose on your lab handout. After 3 minutes, I wanted to measure how well my lactase enzyme was able to break down lactose into glucose and fructose. How do I do that? These are glucose test strips. There's a tube full of strips that look like this. It's a white strip, and it has a green marker, like an aqua green marker at the end of it. I took my test strips, and with the help of some forceps, I dunked them in to each solution.
Please, McKenna Ganath, Stephanie Cuba, Ava Allen, Kat Grove, Cole Charleston, Sophia Ketting, and Anna Lauder.
I stuck my glucose test strips in each test tube, and I moistened the aqua end, and I let it sit for 3 minutes until it dried. When it dried, I checked this color. I know it's difficult to see against this black background. This color against the key on here. And here's what I found. I found that when I tested just the lactose with no enzyme, the glucose test strip found 0 glucose. The test came back negative. What I found when I tested the lactose, the milk, with the lactase enzyme, I found a positive result. That is, lactose was broken down into glucose and fructose. This test strip changed color indicating the presence of glucose, which means the enzyme must have been doing its job.
Please report to the main office.
What you will do in this lab is explore a scenario where you are changing something. I'm not going to tell you what, but you will change something that will influence the amount of glucose produced. So you will produce an experiment that tests a variable of your choice. You will collect data, run the lab, and then analyze the data which you collected. Feel free to watch this video as many times as you'd like. Good luck.